Welcome students. In today's video uh, we are going to describe or discuss from the lesson molecular basis of inheritance one very important topic, that is the properties of genetic material. In our previous video we had discussed regarding the quest for the search of genetic material. We had discussed Griffith's experiment, thereafter the experiment performed by Avery, MacLeod and McCarthy, and at last and the final one was the experiment performed by Hershey and Chase. From the experiment performed by Hershey and Chase, it was evident that the viral DNA had transformed bacterial DNA, and so DNA was equivocally declared and accepted as the genetic material. But some of the viruses contain RNA instead of DNA, so RNA also is the genetic material. Following are some of the important characteristics of genetic material. It should have the capacity to make its exact copy. The another important characteristic of genetic material is it should be structurally and chemically stable. It must be mutated so that the process of evolution can be made possible. It should be able to express itself in the form of Mendelian characters. These are some of the important characteristics of genetic material. Now, the very important thing that we must discuss, that is the difference between DNA and RNA. DNA and RNA both are commonly known as nucleic acid, and both these master molecules are considered as the genetic material also, but we must know what are the differences between the two. DNA is double helical, whereas RNA has only one strand with some exception. DNA has deoxyribose type of pentose sugar, but RNA has a ribose type of pentose sugar. DNA has thymine as one of the nitrogenous bases, whereas RNA has uracil instead of thymine. And DNA can make the copy of RNA by the process of transcription, but RNA can make DNA by the process called reverse transcription. RNA directly forms protein, whereas DNA is dependent on RNA for the protein synthesis. RNA is chemically more reactive as compared to DNA. These are some of the differences between DNA and RNA. Now, you must be remembering that we have come across with the term transcription during the course of our discussion. The process of copying information from one of the strands of DNA into RNA is termed as transcription. Base complementarity between DNA and RNA strand governs the process of transcription, and the mediating enzyme is DNA dependent RNA polymerase. If one of the strands of DNA would copy two different enzymes, it would complicate the genetic information transferring machinery. Genetic transfer of genetic information we have already discussed in our previous discussion in the topic central dogma of molecular biology, where you have seen that the genetic information contained in the DNA molecule is directly transferred into DNA molecule when the DNA is getting copied. That is the exact copy of formation of exact copy of DNA by the process of replication. The genetic information from DNA also travels, or it is transmitted from DNA molecule to RNA molecule when the process of transcription takes place, that is the copying of messenger RNA from DNA, and from RNA molecule by the translation process the polypeptide or the protein molecule is formed, which is the chemical manifestation of genetic information. So, we shall discuss this process of transcription in detail in our next video. Thank you.